That's really the best way to frame it, an arms race between human medicine and the microbial world.
We're talking about antimicrobial agents, AMAs,
Yeah.
and the terrifying global phenomenon they've led to, antimicrobial resistance, or AMR.
Exactly. And for you listening, we want to frame this not just as, a problem for hospitals. It's a crisis of evolution. It's an environmental management crisis.
And we're going to get into the molecular mechanics. We've got a fantastic set of microbiological sources here.
We do.
Our mission today is to really understand, quickly and thoroughly, how these life-saving drugs actually work. And then the big question, why are they stopping?
And what do those high-stakes terms really mean? When you hear a doctor talk about MDR or XDR.
Multi-drug resistance, extensively drug resistant.
Right. We need to understand what that actually means when a patient's life is on the line.
So our sources, they're intensely focused, right? We're narrowing the scope.
Very focused. We're zeroing in specifically on antibacterial agents. So we'll define them, we'll classify them, and we'll detail their 5 primary mechanisms of action.
And then the pivot, the urgent problem of resistance.
Which is really the core of it.
Yeah.
We'll get into acquired versus intrinsic resistance. And this matters right now, today, because the emergence of this resistance is turning what used to be trivial infections into prolonged, complex, and sometimes lethal diseases.
And the cost, not just in lives, but the healthcare costs are just skyrocketing globally.
Absolutely. It's a massive problem.
All right, let's start at the very beginning, part 1, defining the tools. What even is an antimicrobial agent?
Well, at its most basic, the definition is simple. It's any agent that kills or inhibits the growth of a microorganism.
Kills or inhibits, okay.
But that's where the classification immediately has to start, because you have different agents for different pathogens.
Yeah, so you got your antifungals, your antivirals.
Antiparasitics, exactly. But for today, because of the urgency of the AMR crisis, we are focusing strictly and only on the antibacterial category.
Now, I found this really interesting in the source material. Historically, there was this very clear line drawn between an antibiotic and a chemotherapeutic agent.
There was a very sort of romantic distinction, as you said.
But that line has blurred.
It's almost entirely academic now. The classical definition of an antibiotic, it refers specifically to a natural substance, something produced by another microorganism out in nature.
Like penicillin.
Penicillin is the textbook example, of course, from the penicillium mold, but also agents from soil bacteria, like Bacillus species or Streptomycetes.
And the microbiology here is just fascinating. The sources say these are low molecular mass compounds, and they're often produced late in the growth cycle.
Right, during the late stationary phase. And what that tells you is they aren't essential for the microbe's own day-to-day metabolism.
So what are they for?
They're weapons. They are biological weapons used in the fierce, competitive world of the soil or the gut. It's one microbe's way of fighting off its neighbors for resources.
And then on the other side of that old line, you had the chemotherapeutic agents.
Which were defined as anything entirely synthesized in a lab, pure chemistry.
But we got better at chemistry.
We got much, much better. We learned to take those natural antibiotic structures, tweak them, modify them, and often just build them from scratch. We created semi-synthetics.
And that's what erased the line.
That's what merged the terms. Because so many natural antibiotics are now made or modified in a lab, it's just simpler and more accurate to use the single unifying term, antimicrobial agents.
It focuses on the purpose, not the origin.
Exactly. But there's a distinction that is far more critical than origin. And that's the classification by effect.
Cidal versus static.
Cidal versus static. This is paramount in any clinical setting.
And this is something that, if you're a patient, this is a distinction you really want to understand. So let's break it down. What's the fundamental difference?
Bactericidal agents, as the name implies, they kill the bacteria. The action is rapid and it's irreversible.
How do they do that?
They generally go for the big structural targets. Most often, they attack the bacterial cell wall. If you inhibit the formation of that wall, the cell basically bursts. It's a definitive, powerful strike.
And some examples would be?
The beta-lactams, so penicillins and cephalosporins and also vancomycin, all classic bactericidal use.
Okay, so that's the sledgehammer. What about the bacteriostatic agents?
These agents work differently. They don't kill, they prevent growth. They basically shut down the factory.
So they stop the bacteria from multiplying.
Exactly. They inhibit processes like DNA replication or protein synthesis. But the key is that their action is reversible. If you were to remove the drug, the bacteria might eventually start growing again.
And examples there?
Think tetracyclines, chloramphenicol, and the sulfonamides.
So if the static drugs just freeze the population size, they don't actually kill the bacteria. That brings up a really important question. What about patients with a compromised immune system?
That is a very, very sharp question.
Yeah.
And yes, absolutely,
It seems like a huge risk.
it is. And that's the crucial clinical link. Bactericidal agents, because they destroy the pathogen outright, are often preferred for immunocompromised patients, someone on chemotherapy, for instance. You don't need to rely on the host's body to help out.
But the static drugs do.
The static drugs work with the host immune system. They buy time, they hold the line, stop the infection from getting worse, and that gives the patient's own immune system a chance to come in and clear out the remaining bacteria.
So if that immune system is failing, relying on a static drug is a huge gamble.
A very dangerous gamble. The choice is incredibly nuanced.
Our sources actually give us a way to mathematically define this, which I found really helpful. The MBC-MIC ratio.
So let's define those terms for you. MIC is the minimal inhibitory concentration.
That's the lowest amount of drug you need to stop the bacteria from visibly growing in a lab test.
Correct. And MBC is the minimal bactericidal concentration. That's the amount of drug needed to actually kill 99.99% of the bacterial population.
And the ratio of those two numbers tells you everything.
It tells you the drug's character. If the MBC-MIC ratio is less than or equal to 4, we classify the drug as bactericidal.
Meaning the dose you need to stop growth is very close to the dose you need to kill?
Precisely. You can trust that the drug concentration in the patient will actively clear the infection. But if that ratio is greater than 4...
It's bacteriostatic.
Yes. If your MIC is, say, 1 microgram, but your MBC is 8 or 10, that tells you it takes a dramatically higher concentration to kill than to just inhibit. And that immediately suggests you're going to be relying heavily on the patient's own immune function to finish the job.
And the sources add another layer of complexity here. Some drugs can actually shift between cidal and static, depending on the dose.
They can, which makes dosing so incredibly critical.
Okay, so we know how they act. Let's quickly categorize them by who they hit. The spectrum of activity.
We've got 3 main categories here. First is narrow spectrum.
The sniper rifle.
That's a good way to put it. These agents are highly precise. They're active against a very selected group of bacteria, maybe just gram-positives or just certain gram-negatives, but not both.
And the benefit there is you minimize collateral damage to the patient's good bacteria, the microbiome?
Exactly. Then you have the big guns.
Broad spectrum.
Right. These are effective against a huge range of bacteria, hitting both gram-positive and gram-negative species. They're often used empirically.
Meaning, when you know there's an infection, but you don't know the exact bug causing it yet?
Precisely. You hit it hard with a broad-spectrum drug while you wait for the lab cultures to come back. And you'll also see the term extended spectrum used, which is pretty much interchangeable.
But the goal is always to narrow it down if you can.
Always. That process is called de-escalation. Once you identify the specific pathogen, you switch to a narrow spectrum agent to reduce the overall selection pressure and, lower the risk of creating more resistance.
Which is the perfect segue. Let's get into the targets. Here's where it gets really interesting. If a bacterium is a complex fortress, our sources identify 5 primary targets, 5 critical vulnerabilities that our drugs exploit.
And this is the heart of what we call selective toxicity. Finding a target in the bacteria that doesn't exist or is very different in human cells.
Let's start with target 1, the outer shield, the cell wall.
This is probably the most successful target in the history of antibiotics.
Why is that?
Because the bacterial cell wall is made of a unique substance called peptidoglycan. Human cells do not have it at all. It's a perfect target.
So you can attack it without worrying about harming the patient's own cells.
Exactly. And inhibitors that target the cell wall are almost always bactericidal. Because if you weaken that wall, the internal pressure of the cell, the turgor pressure, causes it to just lyse. It bursts and dies.
And the most famous family of drugs that does this is, of course, the beta-lactams, penicillins, cephalosporins.
With the peanut as well.
How exactly do they do it? How do they stop the wall from being built?
It's a beautiful piece of molecular mimicry. The final step in building that peptidoglycan wall is cross-linking all the chains together. And that's done by an enzyme called a transpeptidase, which we now call the penicillin binding protein, or PBP.
The PBP, okay.
The beta-lactam ring structure in the antibiotic looks almost identical to the molecule that the PBP is supposed to bind to, so the enzyme gets tricked.
The drug is a counterfeit key.
It's a counterfeit key that gets stuck in the lock. The PBP binds to the beta-lactam, but the binding is irreversible. It permanently deactivates the enzyme. Construction stops, The cell bursts. It's brilliant.
So clever. Now, another group, the glycopeptides, like vancomycin, they also target the cell wall, but they do it differently.
They do. They're also bactericidal, but their approach is different. Instead of inhibiting the enzyme, they physically block the building materials.
How so?
Vancomycin binds directly to the building blocks themselves, specifically the diala side chains of the peptidoglycan precursors. It's like putting a cover over the bricks, so the brick layer of the PBP enzyme can't even get to them.
And that different mechanism is clinically crucial.
Absolutely. It's why vancomycin is the drug of choice for MRSA, where the bacteria have changed the lock, the PBP enzyme itself. Vancomycin just bypasses the lock and goes for the bricks.
Okay, that's the outer wall. Let's move inside the fortress. Target 2, protein synthesis, the factory floor.
Right. So here, the drugs interfere with the bacterial ribosomes, which are the machines that build all the proteins.
And again, this works because of selective toxicity.
It does. Bacterial ribosomes are a different size and structure than ours. They are 70S ribosomes made of a 30S and a 50S subunit. Human ribosomes are 80S. That structural difference is the target.
So let's look at the drugs that hit the smaller 30S subunit. The sources split them into cytal and static.
Let's start with the cytal ones, the aminoglycosides, like gentamicin. They bind irreversibly to that 30S subunit.
And they don't just start production.
No, it's more sinister than that. They cause the ribosome to misread the mRNA genetic code. So the factory starts producing faulty, non-functional proteins, gibberish protein.
And the cell fills up with junk and dies.
Rapidly, which makes them key weapons against serious gram-negative bacteria, like Pseudomonas.
Now contrast that with the tetracyclines like doxycycline. They also hit the 30S subunit, but they're bacteriostatic.
Right. Tetracyclines don't cause misreading. They just sit on the 30S subunit and act as a roadblock. They physically block the attachment site for the incoming tRNA molecule.
And the tRNA is what delivers the amino acid building blocks.
Exactly. So if the delivery truck can't dock, the assembly line just halts. The bacteria can't make new proteins, so they can't grow, but the cell isn't immediately destroyed.
Okay, what about the larger 50S subunit?
A few classic drugs here. Chloramphenicol, which is bacteriostatic. It stops the ribosome from forming the peptide bond that links the amino acids together.
So it jams the welding machine on the assembly line.
Perfect analogy. And then you have the macrolides, like erythromycin or azithromycin, also bacteriostatic. They bind to the 50S and physically prevent the ribosome from moving along the mRNA strand. The conveyor belt gets jammed.
That's 2 major systems down. Target 3 is even more fundamental, nucleic acid synthesis, attacking the blueprints.
Right. Stopping the bacteria from copying their DNA or transcribing it into RNA.
And the most important class here, arguably, are the fluoroquinolones. Ciprofloxacin?
Yes. They are DNA synthesis inhibitors, but they don't block the building blocks of DNA. They go after the enzymes that manage the physical structure of the DNA.
DNA gyrase and topoisomerase defeat.
The very same. These enzymes are essential for unwinding and rewinding the supercoiled bacterial chromosome during replication. The fluoroquinolones lock up those enzymes.
And what happens then?
The DNA strands fracture. The cell can't divide. It's a lethal blow.
Then you have drugs that are even more direct, the DNA damagers.
These are the chemical sledgehammers. Compounds like metronidazole. They don't target a specific enzyme. They get metabolized inside the cell into reactive intermediates that just physically damage the DNA molecule itself. They're often very effective against anaerobic bacteria.
And finally, you can stop the blueprints from ever being read in the first place.
And that's the job of the rifamycins, like rifampicin. It's a cornerstone of tuberculosis treatment. It inhibits the enzyme RNA polymerase.
So it stops transcription, DNA to RNA.
Exactly. If the cell can't read its blueprints, it can't make any proteins, the whole system shuts down.
Okay, 2 more targets. Target 4, metabolic pathways.
This is a really clever strategy. You find a biochemical pathway that is essential for the bacteria, but doesn't exist in humans.
And the classic example is folic acid synthesis.
It is. Folic acid is vital for making DNA building blocks. Humans get it from our diet. We have to eat it. But many bacteria have to synthesize it internally from scratch.
Which gives us a perfect, unique target. And this is where we see that synergistic action.
Yes, the combination of sulfonamides and trimethoprim. They're often given together as cotrimoxazole.
And they block 2 different steps in the same assembly line.
Precisely. Sulfonamides block an early step, and trimethoprim blocks a later step. Hitting the pathway at 2 points at once overwhelms the bacteria's ability to compensate. It's a beautiful example of synergism.
Okay, last one. Target 5. Just physically destroying the boundary. the cell membrane disruptors.
These are often our last resort agents, partly because the bacterial cell membrane isn't that different from our own, so toxicity can be an issue.
Right.
The polymyxins, and especially collicin, are a key example. They are used against very serious gram-negative infections. They bind to the outer membrane and just disrupt its integrity, causing the cell contents to leak out.
And colistin is a drug that was shelved for years because of toxicity, right? And now it's back.
It is. Its toxicity to human kidneys and nerves meant it was abandoned. But as resistance to everything else has climbed, it's been forced back into service as a drug of last resort. It shows you how desperate the situation is.
And for gram-positives.
You have dactomycin. It's a lipopeptide that inserts into the cell membrane and forms channels, causing leakage and depolarization. It essentially short-circuits the cell, leading to very rapid bactericidal death.
So 5 distinct, sophisticated ways to attack this fortress. It's a formidable arsenal.
It is, but bacteria are the masters of adaptation.
Which brings us to part 3, the threat. Antimicrobial resistance. All those tools we just detailed are failing, and we need to understand how.
Right. So AMR is when a microorganism develops the ability to withstand a drug that was previously effective against it.
And to really get this, we have to differentiate between 2 fundamental types of resistance: intrinsic versus acquired.
This is a crucial distinction. Intrinsic resistance is innate. It's a natural, predictable characteristic of a bacterium. It was never susceptible to that drug in the first place.
Like a gram-negative bacterium naturally resisting vancomycin.
That's the textbook example. Vancomycin is a huge molecule. The gram-negative outer membrane acts like a physical barrier, and the drug simply can't get in.
So it's a known limitation. A clinician can plan for that.
Exactly. It's not the thing that's keeping us up at night. The real global crisis, the one we are truly fighting, is acquired resistance.
This is the scary one.
This is the scary one.
Yeah.
This is when bacteria that were formerly susceptible to a drug gain the ability to resist it. This is usually done by acquiring new genes. And this accounts for the vast majority of the AMR crisis.
And this leads us to the engine that drives this whole process: selection pressure.
It's Darwinian evolution on hyperspeed.
Explain that for us. How does it work?
It's brutally efficient. Imagine a population of 1 million bacteria. Maybe one of them, just by random chance, has a mutation or a gene that makes it slightly resistant to an antibiotic.
1 in a million.
Right. Now, you introduce the antibiotic, it wipes out the other 999,999 susceptible bacteria.
Leaving just that one resistant bug.
And now that one bug has no competition for food, for space. It thrives, it reproduces exponentially, and within a very short time, the entire population is made up of its resistant descendants.
We are creating an artificial selection event.
An intense one.
Yeah.
And the key cause, the thing that is pushing the accelerator on this evolutionary engine, is consistent across all our sources.
Overuse and misuse of antimicrobial agents.
It is the single most important factor driving the development of acquired resistance worldwide.
And we really need to dig into the drivers of that misuse. It's not just about a patient not finishing their prescription. The numbers in our sources are staggering. In the U.S., something like 30% of use is small-scale clinical misuse.
But 70% is large-scale agricultural practice.
70%. Let's start with the clinical side first, though. How does that misuse happen?
It happens in a few ways. A physician might prescribe an antibiotic for a viral infection, like a cold, where it will have absolutely no effect.
But the patient demands something.
Exactly, or they might use a broad-spectrum drug when a narrow one would do, or use the wrong dose. And on the patient side, yes, it's stopping the course early when you feel better. That leaves the strongest, most resistant bacteria alive to multiply.
Okay, now let's tackle the elephant in the room, the 70%. Agriculture. How are antibiotics being used in such massive volumes in food production?
For 3 main reasons. One is for treating actual diseases in livestock, which is legitimate. The second is prophylaxis preventing disease, especially in crowded industrial farming conditions.
And the third, and most controversial,
is as growth promoters. For decades, low subtherapeutic doses of antibiotics have been added to animal feed.
Not to treat a disease, but to make the animals grow faster.
Correct. The mechanism isn't fully understood, but it's thought that it alters the gut microbiome in a way that allows the animals to absorb nutrients more efficiently. It's an economic tool.
So you have billions of food animals receiving constant low doses of antibiotics. That sounds like a perfect recipe for resistance.
It is the perfect incubator, and this leads directly to environmental spread.
So how does a resistant bacteria from a cow on a farm end up causing an infection in a human in a city?
Through contamination. The animal waste, the manure, is full of resistant bacteria and residual antibiotics. That waste is often used as fertilizer or sits in these huge lagoons.
So it gets into the soil and the water.
Exactly. Seepage from these agro-industrial plants contaminates surface water. And the soil, as our sources describe it, is a perfect breeding ground for genetic exchange.
A marketplace for resistance genes?
That's a great way to put it. You have environmental bacteria meeting human and animal pathogens, all in this antibiotic-stressed soup. And this is where the resistance genes can jump species.
Which is the critical next step. How do the genes themselves spread? The sources distinguish between mutational and transferable resistance.
Right. Mutational resistance is slow. It develops from a spontaneous random change in the bacterium's own DNA. This usually gives it low-level resistance to just one drug.
And the classic example is tuberculosis.
Exactly, M. tuberculosis. And we can overcome that slow mutational process by hitting it with combination therapy.
Right, 4 or 5 different drugs at once. The bacteria can't mutate fast enough to fight them all off.
Correct, but that strategy does not work against the bigger threat, transferable drug resistance.
This is the game changer.
This is what drives MDR and XDR. This is the rapid acquisition of high-level resistance to multiple drugs at the same time.
And it's not happening through mutation, it's happening through gene transfer.
Specifically on mobile genetic elements called plasmids, particularly R plasmids resistance plasmids.
And one of these R plasmids can carry the genes for resistance to 3, 4, 5 different classes of antibiotics.
All on one convenient transferable package.
And the main way they transfer this package is a process called conjugation.
Yes. And we should really describe this for you because it's incredible. Conjugation is the closest thing bacteria have to sex.
Okay.
A bacterium that has the R plasmid grows a tiny tube called a pilus. It uses that pilus to physically connect to a neighboring bacterium, which could even be a different species.
And then what?
It makes a copy of its R plasmid and sends that copy across the bridge. In a matter of minutes, a totally susceptible bacterium can become resistant to a whole arsenal of drugs at once.
And because it's a single package of genes, our multi-drug therapy trick doesn't work.
It cannot be overcome by simple drug combinations. It's a quantum leap in the bacteria's defensive capability.
Okay, so that's how they get the weapons. Let's look at the weapons themselves. Part 4. The bacteria's counterattack. What are the specific mechanisms?
Our sources lay out 4 key pillars of defense. 4 strategies bacteria use to evade all those drugs we talked about.
Let's start with mechanism 1, just keeping the drug out. Decreased permeability.
This is mainly a strategy for gram-negative bacteria, which have that protective outer membrane. They can actually modify the little protein tunnels in that membrane, the porin channels.
How do they modify them?
They can reduce the number of them or change their size or their shape to selectively block certain antibiotics from ever getting inside. It's like making the windows on the fortress smaller.
And this works against which drugs?
We see this in really dangerous pathogens like Pseudomonas and Klebsiella. It can block entry for carbapenems, aminoglycosides, quinolones. It's a very effective first line of defense.
Okay, but what if the drug does manage to sneak in?
That brings us to mechanism 2. They just throw it right back out. This is done by influx pumps.
It's an active process.
A very active, energy-intensive process.
Yeah.
These are complex protein pumps in the bacterial membrane that recognize the antibiotic molecule as soon as it enters, grab it, and pump it back outside.
So the drug never reaches a high enough concentration inside the cell to do any damage.
Never reaches its MIC. Exactly. E. coli uses these against tetracyclines. Staphylococcus uses them against macrolides. It's a very common and versatile defense.
All right, mechanism 3 is the most direct enzymatic inactivation. They don't block it, they don't pump it out.
They destroy it. It's a direct chemical counterattack.
And the most famous example of this has to be the beta-lactamase enzyme.
Absolutely central to the entire AMR crisis. These are enzymes that bacteria produce that find the beta-lactam antibiotic, and they chemically break its core structure, the beta-lactam ring.
So they snap our weapon in half before it can be used.
They render it completely inert. And because the genes for these enzymes are so often on those transferable plasmids, they have spread like wildfire through the bacterial world.
And we see this with other drugs too, not just beta-lactams.
Oh, yes. There are aminoglycoside-modifying enzymes. There's chloramphenyl acetyltransferase. For many of our major drug classes, bacteria have evolved a specific enzyme to destroy it.
It's an incredible chemical arms race.
Go.
Okay, the fourth and final pillar of defense, modifying the target site.
The locksmith strategy. If you can't destroy the key, just change the lock.
So the drug gets into the cell, it finds its target, but the target has been altered, so the drug can't bind anymore.
Exactly, and we have to talk about the two critical examples here. First, the one everyone should know, MRSA, methicillin-resistant Staphylococcus aureus.
How does MRSA defeat our most powerful beta-lactam drugs?
It acquires a gene called a mecagene, and this gene codes for a brand new, altered penicillin-binding protein, a new lock,
PBP2A.
PBP2A. And this new PBP2A is a slightly different shape. So the beta-lactam drugs, which were designed to fit the original PBP perfectly, can no longer bind effectively.
So the whole class of drugs becomes useless.
The entire beta-lactam family, from penicillin to the most advanced carbapenems, is rendered ineffective.
And the second major example, vancomycin resistance.
Vancomycin resistance in enterococci, or VRE. This is another target modification.
So if you remember, vancomycin works by binding to the diala part of the cell wall building blocks.
Right. Well, VRE acquires genes that allow it to change that target. It substitutes the final D-alanine for a D-lactate.
A tiny chemical change.
A tiny change that completely prevents vancomycin from binding. It makes the drug useless. It's an incredibly subtle but effective defense.
So those are the four pillars. And the misuse of antibiotics is just the evolutionary pressure that makes these defenses more and more common.
It selects for them, and it encourages their spread.
Which brings us to the consequences. Part 5, the escalation of resistance. This is where we define the threat levels, MDR, XDR, and PDR.
Right. These are formal classifications used by agencies like the CDC to standardize how we talk about the severity of a resistant infection.
It's about how many tools we have left in the toolbox. Let's start with the first level, MDR, or multi-drug resistance.
An organism is defined as MDR if it's resistant to at least one agent in 3 or more different antimicrobial categories.
So, for example, it might be resistant to a beta-lactam, a fluoroquinolone, and an aminoglycoside.
Exactly. This already complicates treatment significantly. It forces clinicians to move to second or third line drugs, which are often more toxic and more expensive.
But it gets worse. The next step up is XDR, extensively drug resistant.
XDR is a massive clinical problem. This is defined as resistance to at least one agent in all but 2 or fewer antimicrobial categories.
So if there are, say, 10 families of antibiotics that could possibly work, and the bug is resistant to 8 of them,
then you're dealing with an XDR infection. You have only 1 or 2 options left. The risk of treatment failure is incredibly high.
And then there's the end of the line, PDR, pan-drug resistance.
PDR is the nightmare scenario. It means the organism is resistant to all available agents in all antimicrobial categories.
So there are no drugs left.
There are no drugs left in the formulary. The infection is, for all intents and purposes, untreatable with our current antibiotics. Patient survival depends entirely on supportive care and their own immune system. We are back in the pre-antibiotic era for that patient.
To really see this escalation in action, we have to go back to the beta-lactamases, because the evolution of this one enzyme family tells the whole story.
It really does. You can trace the stepwise collapse of our best drugs by looking at these enzymes. It starts with ESBLs.
Extended spectrum beta-lactamases, what do they destroy?
ESBLs make an organism resistant to all the penicillins, and also the first, second, and third generation cephalosporins. That's a huge chunk of our arsenal.
But there's still hope.
There is. They're usually still sensitive to our last resort family, the carbapenems. And critically, this resistance can often be overcome by using a beta-lactam drug plus a beta-lactamase inhibitor.
Like clavulanic acid.
Exactly. The inhibitor acts like a decoy. It sacrifices itself to the ESBL enzyme, allowing the actual antibiotic to get through and do its job.
So we have a counter move for ESBLs, but then the bacteria evolved the next level, AMP-C beta-lactamases.
And AMP-C is a much more serious problem. The resistance spectrum is broader. On top of everything an ESBL resists, AMP-C producers are also resistant to another class called the cephamycins.
They are still sensitive to carbapenems, though.
Typically, yes. But here's the critical detail. That standard beta-lactam plus inhibitor combination, it doesn't work against AMP-C.
Why not?
The structure of the AMP-C enzyme is different. The standard inhibitors like clavulanic acid just don't bind to it effectively. So our counter move is neutralized. This forces us to rely almost exclusively on the carbapenems.
Which, of course, leads directly to the highest threat level. The bacteria found a way to defeat the carbapenems.
Carbapenemases. When a lab identifies a carbapenemase-producing organism, it's a crisis situation. These organisms are resistant to everything the AMP-C producers are, plus they destroy the carbapenems themselves.
Our drugs of last resort.
Our last line of defense in the beta-lactam class is gone. And again, the standard inhibitors do nothing.
And these are the true superbugs we hear about.
These are the superbugs: KPC, Klebsiella pneumonia, carbapenemase, NDM, New Delhi metallobeta-lactamase. These are organisms that are often MBR or XDR, and they leave clinicians with almost no good options.
This step-by-step evolution of one enzyme family perfectly mirrors the escalating risk to patients.
It's a clear trajectory towards untreatable infections.
So let's try to summarize this journey. We went from the natural origins of antibiotics, these microbial weapons, to the 5 specific targets they hit inside the bacterial fortress.
And then we saw the bacteria's 4-pillar defense strategy: blocking entry, pumping the drugs out, destroying them with enzymes, and changing the internal targets.
And we now understand that our own misuse of these precious drugs is the selection pressure driving this whole crisis, pushing bacteria from manageable MDR to terrifying XDR and PDR states.
It is a profound and frankly humbling illustration of the power of microbial evolution.
Which brings us to our final thought for you. The source material had this one line that really stuck with me. It said that antibiotic misuse does more than just select for resistant bugs.
Right.
It says it actively induces phenomena in the surviving bacteria like, and I'm quoting here, mutagenesis, genomic instability, and increased gene transfer.
That is a staggering implication.
It is. What does that mean in plain English?
It means that when we use antibiotics poorly, at the wrong dose, for the wrong reason, they don't just act as a simple filter, killing the weak. They act as an environmental stress signal.
A danger signal.
A danger signal to the survivors. It tells them the environment is hostile and unstable, and this seems to trigger a response in them to actively increase their own mutation rate, to become more genetically unstable, to speed up the process of swapping those R plasmids.
So our actions are not just selecting for the fittest. We are actively pushing the survivors to evolve faster, to become more innovative.
We're essentially lighting a fire under their evolutionary engine, forcing them to adapt and share adaptations at an accelerated rate.
So the final question for you to mull over is this. What profound responsibility does that place on all of us, clinicians, farmers, patients, knowing that our behavior doesn't just clear the field for resistant bacteria, but might be actively teaching them, pushing them to become the untreatable threats of tomorrow?
A powerful question and a call for stewardship of these incredible medicines. Thank you for joining us for this deep dive.
Until next time.